welcome everyone. MacroMappers Watercooler. We've got three presenters today who are ready to go, and I know will be very passionate and inspiring. So I'm not going to say too much more because we're going to just get going here. At the very end, I'm going to put a survey link in the chat, and it would be great if everyone just took just a few minutes to fill that out. It just helps us with reporting and telling the Globe office all the good things that we're doing. So we appreciate your help on that. And without further ado, Peggy, Lynn, and Sarah, take it away.
MacroMappers, we started this last year, and we're here to invite more people into the data collection.
Oh, now I see it. Okay, so we're going to start with this video from the 30th.
Welcome to MacroMappers, a worldwide globe macroinvertebrate collaborative project. My name is Peggy Folletta, and I am an education specialist, globe partner, and mentor trainer at Elkhorn Slough National Estuarine Research Reserve in California. My partners are Lynn Hare from Arkansas, who's been a globe partner and mentor trainer for years, and Sarah Merzwack, globe project coordinator for Project Mission Earth in Toledo, Ohio. Last year at the Globe annual meeting in Fredonia, a group of participants started talking about how students find macroinvertebrates so engaging, and they are a great indicator of water quality as well. This discussion evolved into the idea of starting a collaborative project of Globe schools around the world having their students observe and research about a local water body and conduct macroinvertebrate surveys and share their results with and a bit of their culture and with their community and each other. We are about to start year 2. On this slide, it shows blue locations are our partners in the project, and the red locations are schools that have submitted Globe data in macroinvertebrate data, but they are not part of our project, and we're hoping maybe they will join us.
So in order to begin, we had to decide who, what, when, where, why, and how. So ages 9 through 18 years of students, grades 4 through 12, have begun the project, and the study includes the hydrosphere, atmosphere, and biosphere. We try to collect twice a year, 6 months apart, but sometimes that is fall, spring, or dry, wet season, or warm, cold, or the highest time of the abundance of macroinvertebrates. Any freshwater body that's accessible to students, like a river, stream, pond, reservoir, even a ditch. And why we study macroinvertebrates, as Peggy said, was because macroinvertebrates tell a lot about the conditions of a water body, and they record the history of the water body. And so it's very important to see not only the critters that are in the water body, but also the chemistry, and to see if those match up. So how do we do this? We use Globe sampling protocols.
Right, and as you can see, this is a worldwide project, and so we had to come up with ways to coordinate everyone. And the first thing we did was create a Google My Map where we could pin users' locations. We also developed a Padlet where people could post information about their locations and their sampling activities and share information with each other. And then we also have a shared photo album where there are all kinds of photos included now. So I'll show you just briefly. Here is our Google My Map, and this is interactive, so you can see you can click on a location. There are users who have entered some photos of their sites. There's one in Bhutan. And here is our Padlet, and the first column of our Padlet gives users some prompts. So join our map, click on their locations, and add more pins to more locations if they need to. Add some information about their school, about their water site, their data collection, the quality of their water, and then tell their water body story. So you can see that these are the members who have thus far contributed to the Padlet. And Peggy has been so kind as to go through and add a climograph to each location, which has become a very interesting thing to see the differences in precipitation and temperatures throughout the year from each location. And of note here is Bhutan, which we had to change the scale, and it's a heck of a lot more precipitation than all of our other locations. And then just very briefly, here is our shared photo album. And you can see that from the beginning when we were at the annual meeting last year in Fredonia, and one of our teachers here drew our logo. That's still our logo today we're using. And then users have been contributing photos of their students and their sites and their activities with macros.
We've had a lot of success, and it's been very exciting in our discussions that we've had a couple times this year with the groups. But there have been some challenges that some of the schools have had listed here. The macroinvertebrates protocol isn't the easiest of all the protocols if you've ever tried it. And so there's a learning curve. And then there's the learning of the Padlet and the Google My Maps technology. And some schools had a little bit of trouble with finding the equipment and materials. And there was a difference in the participant experience levels, some at the very beginning, and some have been doing this for years. And so that takes a little longer in that learning curve. And school schedules versus seasonal collection. Some schools in the world are in session at different times than other schools, and sometimes they're not in session when there's macroinvertebrates. So we've been navigating some of those issues.
And we'll talk about them more. Okay, so we are gonna, you've seen the My Map, you've seen the photo album, and you've seen the Padlet. But the Padlet's sort of the main focus of this project. So let's go to the Padlet for a second.
Okay, so just to reiterate what I said in that color, you see, give us some information, make it a white post about your information about your water site, make it a yellow one, describe your data collecting experience, a green one, your water quality is blue, and tell your water body story, which would include any cultural or recreational or social information you'd like to share about your site. So people have actually been contributing to this, and we hope to get more and more. Okay, I gotta move my Zoom. And then we also have some resources here. There's a drive, and I think there was a question in the chat about sharing resources. So what we could do is put the link to this Padlet into the chat, and then you can find our entire drive right through there, and also look at all the users. This is a school that I've worked with in Ohio, and you can see here some pictures of work that we've done. And there's a picture of her site, and then some collection activities. So what that teacher did, she actually just collected a bucket of macros this morning, and I joined in with her students and analyzed them and kept. So
Can we look at Uruguay
yeah.
when you're done?
Yes, I was going to. I just wanted
Okay.
to say really quickly, we found is we've done this twice now with her students. So last fall and then again this spring. And then the spring runoff we discovered actually impacted the water quality differently than it was in the fall. And we saw different. So it was an interesting story. If you want to read more about it, there's a nice description that Julie, our teacher, put in there that you can read about. And then I was planning to scroll over. Oh, here's Dario's. So Dario added his information here. Did you want me to try to play his video?
No, I wanted to just kind of go down to, I mean, you've kind of done a good job showing the different pages. So we'll go down, they've done their information about their water site, a lot of pictures, and just continue on down. And then the next one, number 3, let's just go on down.
Okay.
Because what they've done.
Oh, wow. Yeah.
I've done a lot of collecting and go on down. And I mean, theirs is a really thorough and interesting, probably the
Yeah.
most thorough of all. And then we get down to their water quality. They did 2 surveys. They did a Shannon Weaver diversity index, and then they did a pollution tolerance index. And down below, we'll see the results of that in a quick run through these 12 slides, which are, if you click on that,
Click on his link here.
at the bottom, right there, right in the middle. Yeah, right there, anywhere. Should be.
I don't know if it's blocking me from doing this. That's weird.
Oh, it's okay.
Is it doing it?
I don't know.
Slideshow from this post. I don't know what I did. Oh, there it is.
Right, just, quickly run through, just you can see this school on their last field trip, and you'll get the sense of, there's their location, latitude, longitude, continuing on, next page, and they have
There's a video.
Well, it's a video, but we don't need to see it. It's a nice video of their site, and it's really got a lot of water, and it rains there year round. And then the next slide. There are 6th graders that did all of this work. They're 11 years old, and it's pretty incredible.
It's like it's not loading. Hold on. There we go. Students. And their parents too. That's cool.
And they also did it. They've been doing it, I think, for a few years. And you can continue on. And you're looking at some of their invertebrates and their process.
Oh, they're using the clip-on lenses for their phone.
Mm-hmm. Yep. Okay.
That's cool.
Continue on.
Okay. Like they're doing turbidity and pH.
And just to show some pictures of, they did the water quality as well. So moving on, and then we get to their invertebrates, and then he sent his survey to me, and I double checked. We both agreed. So they were surprised to find out that even though it looks so pristine, they really have some contamination in their water, and that's what really shocked this school. So they're probably going to follow up on that. And then they did another, this is the biological monitoring, and this is where it's yellow, and it shows some contamination. So a little bit of surprise for that school. Okay, I think we can go back then to slide 5 in the slideshow.
Okay, I'll let Lynn jump back in.
Okay.
We did that one. Next. Okay. So some of the MacroMapper teams have done it, like we said, a great job. Others really haven't even started. Just a difference in experience and time. It's the GLOBE protocol. If you have a multi-site habitat site, you're supposed to do 20 samples. That's hard. That's no one that I know has done 20 samples. The school doesn't have, many of the schools don't have necessary equipment, and there's other logistical problems. So in year 2, we are suggesting some options. If you're conducting the GLOBE protocol as before, we're really happy because once you get that data, you can do anything with it. And you'll find out in a few minutes that we're seeking some funding to send some equipment kits to those who are struggling. And we will provide the team leaders also with an optional step-by-step process to improve our guidance of them to get them going. Maybe conduct a simpler activity like a pollution tolerance index. And here on the left is the regular GLOBE data entry sheet. And again, fine. It's better if you can do that. If not, the middle one is an example of a pollution tolerance index from let's see, New Jersey Audubon, and this is, you know, there's several of these pollution tolerance indexes, and we're going to look at a video and see how this index is done. And so go ahead, Lynn.
Wait for you to be ready to observe them. Once you finish scooping, you can transfer the macroinvertebrates from the big tray into some of the smaller containers for closer observation. You can see I've already put one in the yogurt cup there, and I've got my ice cube tray over here. Make sure that you have water in any container.
And they're going to put a macro into the ice tray. Ice trays are great because they have lots of slots, and you can put the different types in the different slots. And that's what they're doing. And now they're showing one and saying, here we are looking at it close. Now we're going to identify it. Her words don't seem to be coming through. At least I don't hear them.
Have a tail.
There we
How
go.
does it move through the water? Are there any other characteristics that stand out to you? Now let's look at the dichotomous key to make an identification. Always start at the top, determining if the macro has a shell or no shell. In this case, it has no shell, so we will move down the right side of the key. Noting that it has legs, more specifically 3 pairs of legs, and no wings, will take us down to the bottom row of the key. This macro appears to have 3 tails and feathery gills. It's a mayfly larva.
And now we're going to scroll over to how the index is done.
Macroinvertebrates are indicators of water quality. The presence or absence of certain macros can indicate how polluted a water body is. Riffle beetles, for example, are usually highly sensitive to pollution, meaning they don't do well in polluted waters. So, if riffle beetles are present in your body of water, they could be indicators of good water quality. If you're catching your own macroinvertebrates, keep track of the species you identify and complete the Pollution Tolerance Index datasheet. Let's take a look at the rest of the critters that were caught today. Damselfly larvae are predators. They feed on other aquatic insects, tadpoles, and even small fish. As their name suggests, back swimmers swim through the water on their backs, using their long legs as paddles. Worm-like midge larvae can be found in a variety of colors. The red larvae, like this one, are sometimes called bloodworms. Whirligig beetles have 2 pairs of eyes. One pair looks above the water and the other looks below. This helps them hunt for prey and avoid predators at the same time. Scuds are tiny crustaceans which resemble shrimp. They have 7 pairs of legs and often swim on their sides. Dobsonfly larvae, aka hellgrammites, may resemble aquatic centipedes. They have powerful mouthparts called mandibles, which make them awesome predators. I caught this little fish. It's not a macroinvertebrate because remember, invertebrate means without a backbone, and this fish indeed does have a backbone, but still really exciting to catch. Once you've observed and identified all your macroinvertebrates,
be sure to just gently return them to their natural habitat.
Pause here and practice calculating the pollution tolerance index using the datasheet.
And on the right here is what the results of that survey were. They're all marked and they're all given different values as to whether they are tolerant or not. And based on that, they come up with a score. And in this case, it was a 3.17, and 3s are good. So it's much easier to do, and we want the kids to have success, and we want them out there to see their water bodies. And so even if it isn't Globe Protocol, I mean, I'm a Globe Protocol fiend, but you know, I want kids outside and I want them having a good experience. So we're expanding out a little bit on our project to include some groups that may not be able to do Globe Protocol. Next slide. So this year, we're going to be focusing on the water body stories and the classroom pairing. We didn't quite get to that last time. And so we're asking, I mean, you already saw on the Padlet, which is a key part of our project, that we want to have photos of the students observing, mapping, conducting the surveys, and doing the water measurements. They've already started studying the history of their water body, and part of that is the history of the water is upstream. So if it's a moving body of water, so what's the source? Have there been cultural or social uses of that body of water? You know, a lot of times these kids don't even know the answers to these questions, so they're learning about their own place. But in this case, they get to actually share that with others. And maybe there was a past industrial or recreational or a current recreational use. All that's part of the story. And then what was it that excited or concerned the teams that they found out when they researched their water body? Did these lead to investigative questions or actions? And what were the surprises about the results? Once that's done, we're going to be setting up virtual meetings with teams that have an interest in doing that. And we actually have a meeting with them on Monday, on the 11th, to discuss what's going on at the beginning of this next school year. And so we'll provide the guidance and the Zoom planning with the team leaders for sharing their results. But another option that's always available to anyone in the project is the Padlet, because all the information is there. And they can be as thorough as they want, and they can use the little plus sign and ask each other questions, opening up the notepad and getting to know each other that way, even if they don't do a Zoom meeting. Next slide.
One of the things that we've been talking about is the possibility of writing a proposal to have kits for groups of 4 students, and we're looking at the kit would be enough for teams of 12. So there would be 3 groups of 4 students each, and these are some of the things that we've recommended be a part of that kit. Petri dishes or clear dishes of any kind could be used. Ice cube trays, you've already seen that. Spoons, just any kind of clear or white spoon to help get the critter out of the bucket that you might have brought it into the lab in, or if you're using it outside, then it holds the critters while you're looking at them. Fishnet might be good for both in lab and out in the field. The baby bubbler is something that we have found, and it would be for use in lab study, because you want to keep the water circulating and have air in there. You don't want the critters to die. And so this little baby bubbles is a battery powered bubbler that just adds air to the to the water that the critters would be in. Another thing, the magnifying lenses are really good, the ones especially with the dual magnification. But also this Easy Macro is a rubber band lens that fits onto the camera part of your smartphone, and it's a little more stable than some of the other things that people have used. 1 millimeter grid paper is something that would also be a part of the kit, as well as the ID chart that would have to do with the pollution index. It's not that people can't use their own, they could. And if people already have this or want to get these materials, this is basically all that's required to do the MacroMapper macroinvertebrate protocols. And so we're looking to see about proposal possibilities and getting some kits that, and quite honestly, I think the most expensive part of it will be the shipping. So if anybody has any ideas about shipping these to other countries, please let us know.
You know, that grid paper will be placed underneath the Petri dish for measurement purposes. Because what is it, 1 millimeters?
Yes, it's 1 millimeter.
Yeah, and so that's why that's there.
Yeah, and both of these, if you have this PowerPoint, then these are hot links, and so you can download the 1 millimeter grid paper as well as this pollution index.
I even like the Easy Macro rubber band better than the clip-on for photos. We've gotten better photos with the simpler one. Imagine that. I think that's it. Although,
Peggy, there's our QR code right there to take users to our form to sign up. So if anyone wants to go ahead and join us, they could do that there.
yeah, so you could snap that if you might know some people that might want to join us. We'd love it.
Right, and if you have questions or suggestions, Peggy's email is there, or you can also reach her through the Globe pages, or Sarah or me, any of the three of us.
I think we're open to questions now.
Yeah, what a great presentation. You have one in the chat from Tracy. She wanted to know, what about the DNet?
I did a lot of research on finding the costs of these things. The problem with, I think one of the big problems is the grid size. You know, Globe asks for a 500 micron mesh. And a lot of the things that are sold are not 500 micron meshes. It's much less, I mean, bigger meshes.
Bigger holes.
Yeah, bigger holes. And so people, if they're not using 500 micron mesh, they're not following Globe protocol. But, you know, even in the Globe, I was going to say book, but it's not book anymore. At the Globe site, it does, I think it still shows how you can make a DNet. Is that right, Tracy? Have you looked lately?
We have some information on making a DNet on the resources page.
But again, that, you know, is it 500 micron grid? The cheapest one that I found of, you know, it's DNet. I mean, a DNet or a what do you call it? Gosh, I've been looking at these things until I'm red in the face.
There's a KickNet too.
The KickNet, you can buy one of those at 500 microns, and the cheapest one I could find was $100. And the ones that I've had in the past were student ones, and those are not 500 microns. Those are like 900 or a millimeter. So it's hard to follow Globe protocol because of that right there. Now, I suppose, you know, this is a protocol for using a lot of metadata, saying we used a 900 micron net. We used handheld nets. I don't know very many schools that, I don't know how many schools are really doing this correctly. And so that was my concern. But, you know, use metadata, enter it and say, we use this, this, and this. And at least it's just like the globe weather station. Who has a globe weather station in the right place? Ours was at a, you know, the building was at a 45-degree angle on each side. That's not good, but metadata. So macroinvertebrate surveys are full of the need for metadata, and I'm glad it's there. Does that answer your question, Tracy?
Yeah, I just kind of struggle with that because, I mean, if you're putting a kit together, you know, if you really want people to be sampling according to the protocols.
No,
I mean,
that kit will not be sampling according to the protocols. That way is a kit that's just going to get them out there and seeing. I mean, yeah, it's a struggle.
Yeah. Yeah. I'm just thinking if you, instead of looking to purchase, you know, an actual net, if you can purchase 500 micron fabric and just give them a square kind of thing. And as a part of the kit, just to, you know, I mean, certainly, you know, people can fabricate their own
yeah,
you know, catch system, but
the nylon 500 micron fabric, and it is out there. It, you know, it has a cost. We don't yet have a benefactor for sure, but if we do, that would be the ideal.
Hey, Peggy, what about pantyhose? I wonder what kind of mesh size. I wonder if that would work.
Well, you're going to get a lot of mud in there.
Oh,
too small.
Too small.
Too small.
Probably. Maybe
Yeah,
we could
it's going to get
test
full of
it.
mud.
Do they still make pantyhose?
But that is a good suggestion. But again, because the protocol asks you to go, you know, the DNet has to go in quite a bit. You're going to have all that mud. But if you do collect it that way, see, I've got some metal mesh that's 500 microns, and we could make use a bucket and put that mesh on the bottom and just dump your pantyhose net into that and rinse it. Then you could get the soil out. I didn't think of that.
Oh, that's a good suggestion too. Anne said that brine trip nets for aquariums are close to the 500 microns.
Yeah,
So
that's what we were looking at with the smaller.
Oh, you found some that small? Okay, I wasn't aware that they were that small. Okay. They're still. So I guess what you'd say in the metadata there is if it is close to 500 microns and it is a hand net, you're going to have to describe that your protocol is not using the DNet or the, you know, or the KickNet. But at least I think the grid size is probably pretty important. So yeah, I guess, you know, maybe Jody, you can help us out here and find out which would be a better way to go.
Well, yeah, I'll do some looking into this. It's very interesting. And one of the things, there may be other questions, so I'll keep this short, but you know, we've taken that equipment list that we've put it together, and Haley's been working on putting it into an Airtable so that you can search it. Very, very easy. You can search by protocol, by kit. If it's like an elementary globe kit, we've been working on the elementary globe materials. So perhaps we could make a tag for MacroMappers and put this in there. So I'll put this in my notes. We'll take it to our team meeting, and so then we could also have all your materials listed there for MacroMappers. I'll do a little bit of research on finding some inexpensive 500 micron nets or material or something that could be used for that. And if anyone else knows of any, please send it our way. That would be great. I've got the chat notes here, and we'll go revisit that as well. So thanks for all your ideas.
I'll send you what I have, but I appreciate you doing that, Jody.
Yeah,
Yeah,
great.
and Jody, those little white plastic shallow dishes that you brought when we did the big festival with all the kids, I really like those better than
Yeah,
an ice cube tray because you
yeah.
can spread them out and the kids can all, get around one critter. I think having them share just an ice cube tray, that it's limited spatially, right? You have too many kids
Well,
trying
you
to
can
get
use
around
the
it.
ice cube tray to just put all the ones of the same kind after you look at them. You
Yeah,
look at them and then put it
right.
in the thing.
Hmm.
Or little, I don't know what you use. What's the size of the little containers you're talking about?
They were like what, Julia, like 3 inches square or something like
Yeah, they're
that?
3 inches square. They're white. They're very inexpensive. They're for lab. They're for lab use, like for weighing things out on a scale and so forth. They're
Oh,
lightweight.
those.
They're plastic. They're reusable. They're quite nice, actually.
That
Yeah,
sounds
Good for
good.
soil. Good for soil samples too, Lynn. We use them for soil. Just give everyone a little dab of soil, and
Yeah,
then keep it in
I
the tray.
actually, I'm looking for some. I have some around here someplace. But of course I can't find them now. But we were trying to keep the cost of the kit under $100, and
Yeah.
it was at 60 already, and that didn't include shipping and handling. And yeah, and so we're thinking I don't have any idea about the cost of shipping to certain, that's one thing that we're going to do, but um anyway, yeah, so that's what we were
the
trying to do.
Eslam's supposed to get back to me on that. I've asked him, and
the
he was,
lady in Peggy, the lady in that video mentioned yogurt cups.
Yeah.
So
oh,
users could just take used yogurt cups, clean them out, and even cut them down, and that would
oh,
be
yeah, that's a good
free.
size.
Yeah,
Yeah.
Yeah.
yeah. The trays that I'm talking about literally cost like 3 cents a piece. They're very inexpensive because you can buy them, a big stack of them, and for $10 and have last your whole life probably.
Right.
So,
Well,
so
then maybe
I'll share those with you. I'll look them
we
up
could
and
cut
share.
down on the cost then of some of our other stuff and pay more for the mesh.
Yeah. So, do you have funding now for all the work that you've done so far? This is just you doing it, not you haven't.
We're all donating our time.
Well, it's impressive.
We just think it's worthwhile, you know? You know how Glow people are.
Yeah. Yeah, very similar to that. Gloria, thank you. She put a link in the chat, picture in the chat of a tray. That one looks like a hexagon. Mine are like squares, but same difference. And Gloria's a chemistry teacher, so that's why she knows about these, right?
Cool.
I saw Anna had her hand up for a little while. I don't know if you still wanted to contribute something.
Yeah, I was just going to say this is a really exciting project, and just wanted to touch base with you about somehow wrapping in MacroBlitz, which
Oh,
is really about we went through many of the same issues, trying to make it as accessible as possible. And one of the issues I've found is that teachers are sometimes reluctant because of their confidence and identification. And so taking the photo and uploading it to iNaturalist for crowdsourced verification can help, I
okay.
think.
That's
And
great.
it's if they're already using the lenses and it's just one more thing of somebody having an iNaturalist account and uploading it. And I currently MacroBlitz, the iNat project is only for North and South America because of the challenges of creating things on iNaturalist, you have to be very specific about um, your uh, project requirements and your taxa. So, but I think there's an opportunity if we teach people or show people or sort of connect them into that ecosystem of being able to kind of find what they're doing and get other eyes on what they're observing.
Well,
And
I saw your presentation and it was very impressive. And I could tell you'd been through a lot of trying to get this thing figured out as well. So I applaud what you've done. So yeah, getting together would be great. I'd like to
and
talk more.
it also takes the concern about one of the challenges trying to create a worldwide project is that with the indices and things like that, there can be a lot of variation. Like for example, with the pollution tolerance index, South Dakota's waters, they're in their grasslands will never be more than 3, even though there is no impairment to the water, simply because excellent in all of our pollution tolerance indexes that I have found is you need cold, clear, fast moving water. And the Great Plains just does not have that, except in, very limited places. So it's just something to be aware of, that there is really other valuable data, just the biodiversity aspect from this, that if it doesn't walk over to clear water, or if your chemical, your physical and chemical data is suggesting it's pretty healthy, you have high DO levels, you've got but your aquatic macroinvertebrate population isn't necessarily, you're not finding the Dobson flies, for example. That's a really interesting conversation right there. So anyway, I'll just leave it that, and we can circle back and kind of talk about how we can collaborate.
Well, one more thing about what you're saying is probably, the number of species in good quality water probably varies at different places too. And a lot of the indexes depend on how many different species you find. So that's also, the world out there is a little messy and it's hard to quantify, but those are some of the things. But it heads together.
Well, I love the use of this Padlet. Of course, I work for Jen, right? So, but no, I just think, we don't know where we're moving ahead to with Globe, right? We don't know what's happening with the data, but this shows you that you can still share data with a team. And I love it. I love the climate graph at the top. You can just compare really quick. And if there was, let's say, a Google sheet that everybody attached with from a template that they would all enter data, using the same whatever form of some sort. This is another way to share the data with a smaller group. It's not the world, but it's still really great, powerful. So you've given me some really good ideas about how to organize some of our data for our local region of what we're collecting into a Padlet. And then I'm wondering if you could also add to your Padlet. I'm looking at it right now, but I don't see that. Like all of the materials list could be a column, and then the identification key. I love the dichotomous key. I would love to see that. So maybe it could be right in the Padlet of just everything you need to get going, and then go and add your column with your school. I think that's going to be a really good way for us all to move forward.
I think we need to create a group of just figuring all this out.
Yeah, Peggy, I was going to say that maybe we need to invite some folks to our planning meetings, that would keep brainstorming on this. And Jody, I love those ideas of adding that additional information and a spreadsheet too, a shared spreadsheet to our Padlet. I also realized that our photo album is not linked in the Padlet, so I need to add that.
Oh yeah. Yep.
And I had put,
go ahead.
And just real quick, sorry, Peggy. I had put in the chat earlier that one of our kind of like dream longer term goals is to put together some recommendations for Globe for simplifying and revising the macros protocol. And I think you would be great to help us with that also.
Yeah, but I trying to put a head around this whole thing was, okay, we want it to be Globe because, that's what Globe protocol. Was okay, not everybody can do it. Maybe we have a common set of metadata. Now, we discussed this with Sarah, and her experience with metadata is different than what Globe's is. But for us, it's like what we're not doing that's we're supposed to be. And but anyway, so if everybody had the common set of metadata, at least you could compare, and you could still put it in Globe. You could get your data and you find that people that have the common set of metadata problems. But maybe with this group, we can bypass that and actually do the protocol. I hope.
You know, Ann mentioned iNaturalist, and I think to myself all the time, I love iNaturalist, and I think, you know, how you get verified, it's research grade, you know, it's ready for it's research grade, it's been verified. I'm thinking, wouldn't it be kind of cool if we had that for Globe? Like, so what? Kids are doing MacroMappers and they're collecting it with regular nets, and you know, they're doing whatever. It's still good data. It still could be shared with other schools, and they can use it for school level research. And then we could have a verified research grade for scientists who want to use the data, which means you follow strict adherence to the protocol. And I think a lot of high schools and middle schools even would really want to do that. They'd want to get to that research grade, but shoot, all this work that these young learners are doing is so important to where they're going next. So I'd hate to keep them out of it, right? I'd like to encourage them to collect, like you said, just get them outside collecting data. That's half the battle right there.
Dario's 11-year-olds would do whatever you ask.
Yeah, he's amazing. And that's what I wondered, how you got all that buy-in from those schools. People just jumped on it when you asked them?
Yeah.
That's awesome.
And I'm sure glad to have Sarah and Lynn, because they add so much to this project. And so, yeah, let's be a group.
So Jody, there has been, so the Globe Observer team, not entirely successfully for various reasons, we're trying to do some things that like quality flags with the existing data in order to do some of that checking, so that it's not that any of the data isn't valuable, it's just it's valuable for different purposes.
Yeah.
Didn't get as much, it wasn't really our primary purpose. We started to do some of it. Dave was talking in our technology working group about some things that there's a group with GIO, including Andrew Clark from IGES, Ian, who's also with EDC, I think, and I've forgotten his last name, and somebody else, that are actually doing some quality checks and quality flags. And I think that's a portion of what you're talking about. I think part of that is to go through the existing database and see if you can identify data that just clearly, either one way or the other, either is clearly good or more likely actually, like clearly there are some flaws with this data, you should maybe take it with more of a grain of salt. I like what where you're going with the idea of sort of yeah, like sort of, you know, having a front end. And I think, Sarah, there was wasn't one of the students working with Dr. C trying to look at the Eclipse data and trying to compare, see if there were any differences in the Eclipse data based on whether they were like had fully Globe trained or had just been untrained observers. I don't think he ever got that paper published though. So I think there's a few things that was sort of happening with that. But I think, as with my comment in the chat earlier, I don't think we know what's going to happen in the near term.
Any other questions? We're almost to the end of the hour, but we have a couple minutes left. If anybody has another question for the team or comment.
Well, I guess we've got your names, and we can find you. Make this group.
That's for sure. That's for sure. Yeah, you know, we all read the fine print, right, when we joined Globe, that there's really no out.
Well, okay.
This is not possible.
Yeah.
No. I just put the survey link there. So if everybody take a couple minutes before you sign off, we can keep chatting. But if everyone would take a couple minutes to fill out that survey, that'd be greatly appreciated.
Yeah. And I guess I'll just offer the observation that, you know, the mesh and the protocols and all of that, as you know, it's really to make the data replicable and comparable across sites. But there is a lot of value, even with less than protocol quality rigor, of having historical data sets. So I think, you know, there's absolutely a place for, especially in something like macroinvertebrates, where if you're not going to do it perfectly, but if you're going to do it over time, imperfection over time still yields really good data.
And
there's the thing called crowdsourcing.
For a site taking your observations at your site, you may have different quality needs than somebody trying to do a really rigorous comparison across the globe. And it's not that data has value, just you need to be aware that is it suited for your particular purpose. So I think that's a very good way of stating it.
Well, I've certainly enjoyed this conversation. I know others have too. It's always gets my brain going, and gives me a stepping off point for another idea, another project.